Hemali Phatnani. I'm very happy to be here. I'm jointly appointed at Columbia University, where I'm assistant professor in the Department of Neurology, and at the New York Genome Center, where I direct the Center for Genomics of Neurodegenerative Disease. Within SenNet, I'm the contact PI for one of the tissue mapping centers at Columbia University, where we are mapping senescent cells in different regions of the human brain and spinal cord as it ages. And I'm also the co-chair of the steering committee for SenNet.
What drew you to the field of senescence research?
Well, I started my lab at Columbia about a little over 4 years ago, and I was introduced to Yushin Sir, who had recently moved to Columbia to set up a program on the biology of aging. And this was right around the time that the RFA for SenNet came out. And I had already been working with Vilas Menon, another of our MPIs on this grant. So Vilas and I were interested in applying some of the methods that we've developed and applied to neurodegenerative disease to study the aging brain. And when we were talking to people about whether we should apply for this RFA, they introduced us to Yushin, and we got along and developed this application together, and here we are now. And I could not be happier to be a part of this community. It's a wonderful, wonderful community.
Please give a brief overview of the research you and your team perform.
We are basically at the cusp of adopting new genomics tools as they become available and applying them to problems that have historically been difficult to address. So, for example, if you're interested in looking at how the tissue microenvironment evolves over the course of disease or aging in a way that preserves the spatial context, then you need some of these new methods that have been developed over the past few years. And you need to do this in a way that is scalable, so you can study large numbers of samples and has high throughput. So we like to try out new methods as they become available and figure out how to make them robust and scalable, and then apply them to ask questions that we couldn't have answered without the use of these methods. So, for example, how do we apply these tools to well-designed human cohorts so that we can understand the evolution of pathomechanisms, or we can understand how cellular processes and neighborhoods change as the human brain ages. So for the neurodegenerative diseases that I study, aging is a major factor that plays a role. So if we don't understand how the regions that are affected in these diseases change with age, we'll be missing a huge piece of the puzzle. So we had been interested in studying, applying these methods to the study of the aging brain for some time. And this was the perfect confluence, because some of the phenomena that have been described in neural cells overlap with some of the processes that we see activated during disease. So it's a very interesting biological question, and it's going to be fascinating to see what the intersection is between aging, senescence, and the processes that are active in neurodegenerative disease.
What are your thoughts on SenNet as a community and resource?
SenNet has been invaluable. I was new to the senescence field when I applied, but everybody was open and welcoming, and there are some great mentors in this community. I've worked in many fields through my career. I haven't encountered any as nurturing and open as this community. And I think this consortium itself is very, very special in the kinds of people that it has brought together and the attitudes it has fostered.
How do you think SenNet fits into the broader senescence research community?
There are an amazing set of technological advances that can be brought to bear on this problem. And the field is moving really fast with, um, a lot of new models and a lot of new systems to which these technologies can be applied. So having this consortium formed at this time in the evolution of the field is really critical because we can be nimble and agile and we can work as a community to refine, discuss, and disseminate the findings that we make through applying these new methods. It also fosters the adoption of new tech faster. It does so in an informed way. So we grow as a result of it, and the field grows as a result of these efforts.
In your opinion, what are the greatest challenges to SenNet?
As the pace is picking up, we have to make sure that the rate of data production doesn't outstrip our ability to learn meaningful things and glean biological insights from the data. Now more than ever, it's imperative that we communicate with each other and make sure that we're bringing each other along on the journey. Uh, this is the time for us to make sure that we're actually talking about real biological insights and the implications thereof. As a common fund program, I think SenNet is one of the first that's actually going after uh, real biological questions instead of just geared towards data generation. And it's also going after cells that are relatively rare. So it's it's a very ambitious program, even for a common fund program. It's technologically challenging and it's conceptually unique, I think, or was one of the first of its kind. So in a sense, there hasn't been a roadmap for what SenNet set out to do. And we've been learning as we go, and that's part of the excitement in the community, I think. And it's like we're on this great big adventure together. And I think that brings us together more than anything else could.
What would you tell students and junior investigators who want to research senescence?
I would say be as open as you can. This is a great community. There's no hierarchical barriers. There's everyone's input is welcomed, and the mentorship is one of the biggest strengths of SenNet. The junior investigators have their own working group. They develop programs that they present at the SenNet meetings. They are part of a vibrant community, which I would encourage you to join. I would also encourage you to participate in any working group that you can. Your voices are welcome and your input is highly desired because you are the guys who are actually doing the work at the bench. So this is where development and dissemination and collaboration happens. So participate. It's a great community to be a part of.